Okay, so this morning's speaker is James Lee from the University of Minnesota. He was a Steven Pinker student at Harvard. He's now been at Minnesota Psychology Department for several years. And he knows a lot about statistical genetics, psychology, twin studies, so by all means take advantage of him while he's here.
All right, thank you, David. So I'm going to be talking about a subject called population genetics. This can be defined as the study of how genetic variation is affected over time by various evolutionary forces. And I'll be talking about some of those forces today. So the geneticist Theodosius Dobzhansky once said that nothing in biology makes sense except in the light of evolution. And I happen to think that analogously, nothing in GWAS actually makes sense either, except in the light of population genetics. That might seem like a bold claim, a high bar to meet, so I'll try my best to try to meet it. So first, I'll start off by talking about two evolutionary forces, mutation and genetic drift. So mutation is the origin of all genetic differences. Even if at some point all individuals were genetically identical, equivalent to a large brood of monozygotic twins, it would still be the case that after a while, mutation would cause individuals to differ from each other. Okay? Now, mutation is an incredibly rare event. I've depicted it here. So you can tell that this is a mutation because this allele is not present in either of the parents, actually. And actually, empirically, that's how often mutations are detected. So the probability that a mutation occurs in any given transmission event from parent to child is extremely low, given by that number over there. But in aggregate, they can have a substantial effect. In fact, it's been estimated that new mutations increase the heritability of a typical trait by about 0.001 each generation. Okay? I also want to point out that this diagram happens to illustrate another evolutionary force, recombination. So recombination is, you can sort of think of it as like the parental and maternal contributions to an individual's chromosomes. They sort of break apart at a given point, and then they fuse back together, creating a kind of new gamete that's a fusion of the parental and the two parental contributions. And so this is what we call recombination. It's a bit unrealistic here because here I have two recombination events occurring very close together. That would be extremely rare, but I just have it there to give you the idea. All right, so one question that immediately arises is that if new mutations really increase the heritability of a trait by this much each generation, you might naively expect that every trait then should have a heritability close to 1, because it would not take that much time, even starting from 0, for the genetic variance to become much, much larger than the environmental variance. Okay, so I'll explain why that actually doesn't occur in most traits we look at. The heritability is well short of 1 later on. All right, so if a mutation occurs at a previously monomorphic site, so this is a site where everybody is exactly the same. Everybody has, for example, the genotype GG, say. So if a mutation occurs at such a site, then the frequency of the derived allele, I'll say this a lot, that just means the mutant allele or the new allele that wasn't present before the mutation. The frequency of this in the population has to be 1 over 2n, okay, where n is the population size, all right? So there's n people in the population. Each person actually has 2 bits of DNA at each site, 1 from each parent. So if you have this one new copy that has appeared as a result of mutation, its frequency has to be 1 over 2n. However, in living populations, we frequently observe polymorphic sites where the derived allele has a frequency much higher than that, 0.10, 0.20, and so on. And so a question that immediately arises is how is this possible? Okay. Well, here's one way that can happen. So suppose that the carrier of a new mutation has 2 offspring. Okay. So if the population is staying at a constant size, each individual has on average 2 offspring. So suppose I have this new mutation and I have 2 offspring, okay? Now, by sheer chance, it might be that actually both of my offspring inherit this mutation, okay? So if you think of it as coin flips, heads mean that the mutation goes, tails it does not. It just might be that the Mendelian coin comes up heads both times, and both of my children just happen to inherit this mutation, even though in expectation only one of them should have, right? So in this way, the derived allele has changed in frequency in one generation from where it started, 1 over 2n, to 2 over 2n, okay? Conversely, the opposite fluctuation can occur. Maybe the Mendelian coin comes up tails both times. Neither of my offspring inherits the mutation. And then the frequency goes back to 0, where it was before. So this process, where you have allele frequencies changing across generations as a result of random undirected forces, such as, for example, the randomness of Mendel's first law, this is called genetic drift. Genetic drift, okay? Okay, so here's a graph depicting how genetic drift works. So basically, usually when a new mutation appears, it will bounce around in frequency for a while and then disappear. Okay, that's typically what happens. If the chance fluctuations are large enough in magnitude, it's inevitable that there's going to be a fluctuation that's going to take you back down to 0. Okay. Occasionally, however, genetic drift will take the allele frequency to 0.10, 0.20. In fact, it is even possible, even though this might not be intuitive at first, but it is possible, for the frequency to go all the way to 1, okay? As will probably happen to this mutation, since it's closer to that barrier than to this barrier. So in fact, if you compare the human and the chimpanzee genomes, there are roughly about 30 million locations where humans are 100% for one allele, chimpanzees are 100% for the other. Probably most of those fixations are due in large part to genetic drift. Okay. All right, so genetic drift has played a large role in not only shaping these fixed differences between, say, humans and chimpanzees, but also patterns of genetic variation within each of these species. So for example, there are about roughly 10 million SNPs where both alleles are common. That is, both alleles have a frequency of over, say, 0.01. This varies a bit by population for reasons that I'll explain later. And so basically, the basic story at most of these sites is that at some point in roughly the last million years, a mutation occurred. And initially, the frequency of the derived allele was 1 over 2n, and then genetic drift, for the most part, has taken it to 0.3 or 0.44 or whatever it happens to be today. Okay. Okay, so I have a diagram up here that I'll have occasion to refer to frequently. So this is a diagram from a paper by Simon Gravel and colleagues a number of years ago. So this is basically depicting in a highly idealized and actually in some details incorrect way the population history of Yorubans, Yorubans being a West African population, CEU, a population in the United States, most of whose origin is probably Southeast England, CHB and JPT being Han Chinese and Japanese, who for some reason, some purposes are often just merged. So this is basically saying that in Yorubans, they've been evolving for a long time at a more or less constant population size, roughly 10,000. Roughly 50,000 years ago, some populations split off from the Yorubans. And this branch that departed from Africa, presumably, had an estimated size of fewer than 2,000 individuals. And then more recently, this branch split again with the ancestors of Europeans having a size of about 1,000, and the ancestors of East Asians having a population size of about 550. And then subsequently, as we get closer in time, these two Eurasian populations began expanding extremely rapidly. Okay, so keep this in mind as we step through various phenomena that I'll be talking about. Okay, so to some extent,
yeah. Can I ask you a question? So when you say they're expanding rapidly,
yep.
You mean like effective population, right?
Yes, effective population size, which is not the same as the total number of individuals, but probably is it's often said is typically about 1/3 to 1/10 of the actual number of individuals, empirically.
How would expansion happen other than through like mutations being introduced? Are you going to talk about that? I don't know.
Why does the population start expanding?
Yeah. Why would it expand differentially there than it
Well,
has
the proximate explanation is the invention of agriculture around roughly 10,000 years ago, 12,000 years ago.
Okay, so if a population doubled in size,
Yeah.
that would correspond to an increase in effective sample size too, probably.
Yes.
I see. Okay.
Yep. Okay. All right. Okay, so as a result of this population history, you're going to see some genetic differences between these various populations. When we're talking about differences in allele frequencies, it turns out that drift is the evolutionary force that has made, in the future it's going to be quite different, but up to this point has made quite the largest contribution to these allele frequency differences. For example, if you compare, for example, Europeans and East Asians, allele frequency differences where you're counting, it doesn't really matter, the derived allele, the minor allele, the GWAS reference allele, and some summary stat file. If you look at the difference between Europeans and East Asians, differences of 0.1, 0.2, 0.3, and so on are in fact quite common. So here's a visual depiction of how this comes about. So suppose that you have some ancestral population where the allele frequency at a given SNP started at 0.5. As we go forward in time, these effects of finite population size cause all these squiggles. Now suppose at that point the mother population splits into two daughter populations that thereafter never interact, they never exchange any migrants. What happens then is that the random walk continues, but it just happens independently in the two populations. Okay? So for example, in population 1 and population 2, they might end up going like that. Okay, I actually cheated a bit. I tried this multiple times until they started to go apart. But the thing is, I only had to do that like 3 or 4 times. It's very probable for that to happen. Okay. All right. Now, this is actually the origin, for the most part, of the phenomenon that we call population stratification. So suppose that a GWAS sample is drawn from multiple populations that differ in allele frequency, primarily because of genetic drift, and their phenotypic averages, such as the average chopstick skill or something like this, differs across these populations for reasons that don't have anything to do with genetics, or at least not the SNP that's being tested. Okay? And this is what we call population stratification, all right? Or the so-called chopstick gene, as Patrick referred to it. Okay, occasionally I'll have slides with more technical material that I'm just going to skip, but it may be worthwhile for you to take a look at these later. Okay, so now let me talk about more or less the same phenomena, except, okay, I think I meant to put recombination up there, so that's a typo. I'll fix that later if that turns out to be the case. Okay, so this foregoing account of genetic drift, which I've just related to you, this develops into something called diffusion theory when it's developed more quantitatively. This is now seen as a forward time approach. The reason why is that this contrasts with another approach called coalescent theory, which has developed since about 1980, which is essentially looking at the same phenomena, mutation and genetic drift and so on, but except instead of theoretically, you think about like something in the past that's actually empirically more or less hard to access for us, and then just kind of hypothetically imagine what would happen going forward in the future. Coalescence theory starts with present-day genetic variation. This is something that we can easily empirically measure, and then posits what happens to this as we look backward in time. Okay. Okay. And the reason why I'm going to go into coalescent theory is that it turns out to be an extremely powerful way of explaining many phenomena that we encounter again and again and again in GWAS and other kinds of studies. Okay, so to explain coalescent theory, so focus on a specific segment of your genome. I'm going to just call this like a sequence. So this will be like from my diagram earlier, my toy diagram of mutations and recombinations and so on. So a stretch like that, like suppose like 100 adjacent nucleotides, let's call that a sequence, okay? And so you, at a particular location in the genome, carry two homologous sequences, one inherited from your father and the other inherited from your mother. Now let's concentrate on a particular one of these sequences. Now at this point, think about not only yourself, but another person, okay? This other person also carries homologous sequences in that particular region of the genome. And of course, this person also inherited the two copies of that sequence from the individual's own parents, mother and father. Now, imagining, in your case, following the chain of transmission from you for a particular one of these homologous sequences, from you, suppose it came from your father, from you to your father, to whichever grandparent that sequence came from, and to the great-grandparent, and so on, going back in time. Okay? Now, it'll turn out that, of course, that there is a similar sequence extending back from a similar sequence of genetic transmissions that extends back from the homologous genomic sequence in this other individual we're thinking about. Okay? So suppose this is you, and this represents the chain of transmissions from parent to grandparent and so on. A similar such chain extends back in time from this other individual that we're considering. Okay? Now, at some point in the past, it must be the case that these two chains of ancestry, these two chains of descent, meet or coalesce, the word coalesce meaning join together, in a common ancestor of the two sequences. Okay? So for example, these two meet in their common ancestor pretty soon at this point right here. All right? So that's basically what coalescent theory is about, how a sample of sequences coalesces back to their common ancestor. So to get into it a bit more formally, suppose that we have a sample of n sequences taken from members of the present generation. Now, let's think about the following question. What is the probability that two specific sequences coalesced or descended from the same ancestral sequence immediately in the previous generation? Okay? What is the probability that these two people were considering, the common ancestor sequence was in fact immediately in the previous generation? Okay? And the simplest coalescent model it makes a number of assumptions, such as there's no natural selection, there's no recombination, and so on. Furthermore, let's suppose that the next generation, as we consider this process forward in time, is formed from the current generation by random sampling with replacement. Okay? This is often called the Wright-Fisher model of genetic drift. I think erroneously, because Fisher published it 9 years previously, and Wright actually probably got the idea from him, but whatever. As I said, I'm against terminological legislation, so let's just keep calling it the Wright-Fisher model. Now, this model, as I said, is in many ways unrealistic. For example, it does not recognize the existence of two sexes and various phenomena that come up with that. For example, if you have a sibling, it must be the case that the coalescence event is immediate. But actually, it turns out that for our purposes, these failures of realism are not particularly important. Like, for example, the existence of two sexes perturbs the process in the short term, but then in the long term, it becomes immediately exactly like the standard coalescence. So usually, these failures of realism are not important or don't matter for what we're talking about today. Okay, so here, by that arrow, what I mean by that is I mean that sequence A in the previous generation is the parent of sequence 1. Okay? So this question I'm asking is, what is the probability that the sequences 1 and 2 coalesce into sequence A? That is, what is the probability that A in the previous generation is the parent of both 1 and 2 in the next one? Okay. Well, now suppose that the population size is n, and also that it stays constant across generations. That's one of these unrealistic assumptions, but we're just going to go with it for now. Now, by exchangeability, each sequence in the previous generation is equally likely to be the parent of sequence 2. Okay. So that means that the probability of coalescence is 1 over 2n. Okay? Once I know that sequence 1, this is its parent, then who is this parent of sequence 2? I pluck it at random out of the entire population, and then the probability to land on this has to be just 1 over 2n. Okay? And so that setup suffices to describe the simplest possible coalescent model. Okay? So coalescent models are often depicted as trees like this. The terminal nodes represent the members of the present-day sample. The edges represent, as I showed you in the earlier slide, these chains of descent going back to parent sequence, grandparent sequence, and so on, going back in time. And whenever these edges meet, that represents a coalescence event. Okay? And these coalescence events occur at a rate that's proportional to 1 over 2n. The coalescent rate slows down as we go back in time, because once coalescent events have already happened, there are fewer sort of buddies that you could possibly meet, but there's always this factor of 1 over 2n. Okay? So importantly, that means that in a smaller population, n being smaller, coalescence events happen more rapidly. Okay? That's extremely important to keep in mind. In a smaller population, coalescence events occur more rapidly. Okay? Okay. So let me now talk about mutation viewed in a coalescent framework. So suppose that a mutation occurs at a particular point in time, like, for example, right here. I mean, this might not be obvious at first, but I hope it becomes obvious. It must be the case, then, that all descendants of that mutated sequence must bear that mutation. Okay? That is, that mutation must be carried by all the descendant sequences coming all the way down to the present day. All right? So, for example, in this particular tree, it must be the case that those 3 sample members on the left all carry that red mutation. Okay? So notice that in the sample, then, the frequency of the derived allele is 3 over 5. Okay? And if you were looking at the actual sequence data, it could look something like this. This would be compatible with the tree that we're looking at. Right? Now, if you have a sequence like this, a relatively short stretch of the genome, you may see mutations at more than one site. Okay? So suppose that happens to be the case in this sample. We have a second mutation that occurs right there. Okay? And so basically, the descendants of that branch all have to carry that mutation. And it happens that the sample frequency of the derived allele has to be 2 over 5. All right? And so the actual sequence data in this case might look like that. Okay. Okay, I promise I'm getting to a conclusion here, so be patient. So the edges that connect the terminal nodes to the rest of the coalescent tree, that is, the edge that takes a present-day sample to the rest of the coalescent tree, this is called a tip. Okay? So before any coalescent event occurs involving that sample member, we call that branch a tip. Now, a polymorphic site, a SNP, that occurs only once in the sample, since these are so important, we have a special term for these. These are called singletons. A singleton is the result of a mutation that occurs on a tip. Okay? So if a mutation occurs on, let's call it an interior edge, it has to appear more than once in the sample. However, if you have a mutation occurring on a tip, that will lead to a singleton. All right? So that would be a singleton. That would be a singleton as well. If we happen to have mutations here, those would be singletons also. And so once we update our sequence data to reflect those events, it might look something like this. Okay. All right. So now what can all this machinery about coalescence and mutation, we can use this to explain a number of phenomena that we run into in GWAS. For example, it can explain why we observe the spectrum of allele frequencies that we in fact observe. For example, why is it that we observe some SNPs that have a MAF greater than 0.4 up to 0.5, and then another fraction of our SNPs in our study are in the next interval, and so on. Okay? So let me proceed to explain that. So we've seen that any mutation occurring on a tip results in a singleton, okay? A SNP with a derived allele frequency of 1 over n, where n is the sample size, okay? Now, singletons are actually the most common kind of SNP because the total length of the coalescence tree that's contributed by singletons is longer than that of any other type of mutation, okay? So I actually measured this tree with a ruler that I borrowed from the front desk. And so basically I measured the lengths of all the singletons and then measures the length of all the parts of the tree where if a mutation happened there, it's going to result in 2 over n and then 3 over n and so on. And then 4 over n happens to be impossible in this particular random tree. But so anyway, you see that the total length of the tree where a mutation will lead to a singleton is roughly half. And then it monotonically declines, which is not necessarily guaranteed to happen in a random tree, but that's the expectation. Yep.
Just clarification.
Yep.
You were talking about a sequence.
Yep.
The SNP, because the SNP can have only, so many mutations, right?
So basically, multiple mutations can be present in a given sequence, and you're just counting how many of them are singletons.
Got it. Okay.
How many of them are?
The tree is 4.
I mean, it's just like in our GWAS summary stats file where you have the column for the allele frequency, and then you could just make a histogram of that or something. And you could say how many are here, how many are here, how many are here.
But the tree is for a full sequence. Yeah. All right.
Yeah, I didn't quite hear that, Jonathan.
The tree is tracking a sequence, not just a SNP.
The tree reflects the ancestry of some chunk of the genome that does not experience recombination. And there, in that sequence, there could be multiple SNPs that share that tree. Yep. Okay, so my ruler, I made this tree kind of randomly, so I'm a bit lucky that my ruler measurements actually confirmed this point that is actually, the quantitative expectation that there's a monotonic decline in frequency. The most common type of SNP should be a singleton, and then the next most common type is 2 over n, 3 over n, and so on. Okay. So coalescent theory can be used to actually rigorously prove what this example kind of suggests. This is that the probability that a SNP has a derived allele frequency of p is proportional to 1 over p. Okay, 1 over p being this monotonically declining curve. And we can see how the intuition for that coming from coalescence theory. Now, this is why there are more rare SNPs than common SNPs. So 1 over p, as I said, is monotonically declining. Rare SNPs are things with a p of less than, depending on the purpose, 0.1, 0.05, 0.1. And there's always going to be a lot more there than over some interval of equal size, over to the right. And actually, a more precise statement than this can be made. Because it turns out that the probability distribution 1 over p has this rather neat property, which is that if you take its integral, that means the cumulative probability, over each of these ranges, like going from 0.001 to 0.01, 0.01 to 0.1, 0.1 to 1, these integrals are all equal in value. Okay? That is, according to this neutral theory, each of these intervals of derived allele frequency should have an equal number of SNPs. Okay? Actually, Fisher derived 1 over p way back in 1930. So in principle, you could have predicted then that once the human genome is sequenced 1,000 years from now, we'll be able to measure this. But actually, things move faster than perhaps one would have thought back in 1930. So we can actually look at this now, right? How well does actual empirical genetic data conform to this theoretical prediction? Okay, so this weekend I looked this up. There's a recent paper on bioRxiv by Wayne Stein and a number of other authors. This was actually a genome sequencing study, but it happens in one of their supplements. They provide the number of SNPs in all of these intervals. And so I was interested in seeing how well it goes. And this is what it is. Pretty amazing. Actually, I was at a lab meeting recently where a student was presenting her work on GWAS of tobacco and alcohol use, and she was showing QQ plots of different MAF ranges. And she happened to say, I find it very interesting that there's roughly an equal number of SNPs here and here. And I immediately pointed out, no, that's not surprising at all. And then I proceeded to explain this. And people started to look at me strangely. Well, but now that you know this, you too, at your next lab meeting, when someone brings this up, can explain this and be thought to be a practitioner of spooky black magic.
So James,
Yep.
you're saying there that it's an attempt to partition heritability? This has nothing to do with partitioning heritability, does it? It's
No, this has nothing to do with
it's
the
just
main
counting
purpose
up
of the
the
study.
number of
Yep.
SNPs and
Okay. Actually, this relationship actually breaks down for small MAF. And then for the smaller intervals, it breaks down even worse. I believe that there's something like 20 million SNPs in the next interval. So basically, deviations from the simple coalescent model or the simple Wright-Fisher model can cause deviations, particularly at very low MAFs. For example, bottlenecks, expansions, natural selection, and so on. But remarkably, over the range of MAF where we've concentrated GWAS on so far, this prediction holds remarkably true. Okay, now one question you might be thinking about as you look at these numbers. So we basically use GREML and related techniques to try to estimate the heritability that's due to like this versus this and so on. These numbers where there's more and then due to deviations from the coalescent model, even more SNPs that are rare than common, this might lead you to ask the question, should most heritability be due to rare SNPs as opposed to common SNPs? Okay. So if there's time, I'll actually talk about that later on. Okay. So here's another question that coalescent theory explains. So it turns out that actually the number of common SNPs is always kind of in the ballpark, because actually this depends on what population you're looking at. For example, Africans have more common SNPs than Europeans, who in turn have more common SNPs than East Asians, and so on. Okay. Now it turns out that the explanation of this can be found in coalescent theory, because the African populations, most of them anyway, have traditionally enjoyed a larger effective size without all these bottlenecks, bottleneck being a technical term for a drastic reduction in population size. And so basically, the coalescence trees, for example, in Yorubans tend to have a greater time depth. And with that greater time depth, there's just more opportunities for mutations to create SNPs. Okay, so let me answer another question using coalescent theory. This question being, why is LD between distant SNPs nearly always very minuscule, but between nearby SNPs often high? So this is a figure that shows a color-coded correlation matrix of SNPs on chromosome 22. I think the more red means higher the absolute R. And so you can see that when SNPs are far away, they're virtually uncorrelated. But then if we blow up the diagonal, we can see that often SNPs are strongly correlated with their neighbors. Okay, so here's the coalescent theory explanation of this. So let's start with the fact that recombination events between nearby SNPs occur very infrequently. The probability of recombination between two adjacent base pairs actually is sort of a coincidence. Well, maybe it's not a coincidence. Maybe there are no coincidences. But it's very similar to the mutation rate. So it's a very small number, okay? Although it varies a bit between recombination hotspots and so on. But on average, across the human genome, it's roughly about that number. So this means that this idealization or assumption that we made to sort of derive coalescent theory, which is that there's no recombination, this is in fact pretty much true for SNPs that are close by in the genome. Okay, so recombination scrambling our nice coalescent tree, that very rarely happens for nearby base pairs. Okay, so this means that this came up in Jonathan's question, that it is indeed true that SNPs that are close by in the genome, that the sequence in which they're embedded will in fact share exactly the same coalescent or tree of ancestry. Okay, that is, if a chunk of chromosome has never experienced or very rarely experienced recombination, the ancestral history of all SNPs in that chunk must be the same. Okay. So going back to linkage disequilibrium, let's consider the two SNPs depicted in this example. Notice that they are perfectly correlated. Okay. Their LD coefficient R is equal to 1. Oh, actually, I should back up a bit. So oftentimes, I'm talking about the properties of a given sample, which actually might be quite small, like 5 in this toy example. And then I pass over to properties of an entire population. So this, in coalescent theory, for many purposes, you have to be careful about that leap. But for what I'm talking about today, I think most of the time it's OK. So sample, population, let's just think about it a bit interchangeably. OK. So here, the LD coefficient between these two SNPs is equal to 1. Because we know that if a sequence carries the red mutation, it does not carry the green mutation. Okay? So if I know that C is present in this particular haplotype, I know that at this other SNP over here, that the allele is going to be A. Okay? So basically, we can see that given the placement of the red mutation, any mutation that occurs here or here, which is actually a substantial fraction of the total tree, will produce two SNPs that are perfectly correlated. Okay? And so this is basically why nearby SNPs are often in strong LD. There's no recombination, so they share the same tree. And once you have the same tree, there's a large subpart of the tree where mutations will lead to perfect LD. Okay? Now, this phenomenon has many profound ramifications, including the difficulty of identifying the causal SNP that's responsible for any of these peaks in the Manhattan plot. For example, this comes from schizophrenia. And like any of these peaks you see, these might encompass, in some cases, more than 100 SNPs. Okay? And because of this strong LD locally, it becomes very hard to tell which SNP is actually the causal SNP that's driving that signal. Okay, coalescent theory also explains, although it has to be elaborated a bit, why different populations differ in the extent of linkage disequilibrium. We saw some nice slides of that last week where we looked at, in haploview, how LD blocks tend to be shorter in Africans, a bit longer in, well, much longer actually in Europeans, and then a bit longer in East Asians. And then if we happen to look at American Indians, they would tend to be even longer still. And the explanation of this phenomenon is also because of differences in population size, severe bottlenecks, for example. So what happens in a smaller population, it's actually pretty analogous to why different populations differ in their numbers of SNPs. Basically, in a small population, the coalescent tree will reach its root much more rapidly. And this means that there are just fewer opportunities for recombinations to happen. Okay? So let me try to show that in a picture. So let's ask, we have two SNPs here. And then we might wonder, in this particular population we're considering, are these two SNPs going to show linkage disequilibrium? Okay? So how well will the presence of T predict whether A is there? Okay? Now, if we think about this from the standpoint of coalescence theory, let's think about the tree joining these two sequences, going back in time. Now, it's kind of a race. What happens first as we go back in time? This T and this T coalescing, another mutation making their sequences different, perhaps, or a recombination event. Okay? Now, basically, in a smaller population, coalescence will tend to win, okay, because coalescence is proportional to 1 over 2n. So it becomes increasingly likely as the population size shrinks or it's gone through an extensive period of a bottleneck, small size, that the first thing that happens as we look back in time is, in fact, a coalescence event, okay, rather than a recombination. So if we know that that's the ancestry of the top two, that the first thing that happened to them was coalescence rather than, say, recombination or a mutation, then we know that they have to be identical. Okay, they have to be identical, which means that that they are in fact in LD. Okay, and so this is basically why LD blocks tend to extend longer in these populations in the sequence that I described. Okay. So another way to put it is that if recombination occurs before coalescence, then the two SNPs don't in fact share the same coalescence tree. Okay, and then this argument about the same tree and then mutations here and here will lead to perfect LD doesn't follow. Okay, at this point I'd like to make somewhat of an aside. This aside being, because this picture is actually extremely convenient for making this point, notice that only SNPs that have similar allele frequencies can be in strong LD. Okay, so here I've depicted two SNPs where the MAF is both 3/7, and then you can see that it's possible for them to be in perfect LD. However, if we have two SNPs with different MAFs, like for example over here I've turned it into 1/7, you notice that A at the second SNP invariably appears with T. If we have A, we know that T is present at the first SNP. But then nevertheless, for example, T does not predict A very well. T often still occurs with G. Okay? So basically, if two SNPs have very different MAFs, even if they're as correlated as maximally possible, still it's impossible to partner everyone up in a way such that you end up with a strong R. Okay? And so that's why two SNPs can only be in strong LD if their allele frequencies are similar. Okay? Yep?
Yeah, I was just trying to understand, but there is a directionality to this, right? A is predictive of T in this case. So there is kind of a asymmetry in terms of the conditional prediction.
Yes, and I think the way that shows up is that other measures of LD, for example, I believe D, do capture that, but R does not. But it turns out that R is what matters for a variety of things. Like for example, if you have a hit in a GWAS that is a common SNP, for a while people debated, oh, that just could be because it's an LD with a rare SNP that has a powerful effect. And then people wonder, well, GREML says common SNPs have this heritability, but could it just be that the common SNPs on our chip are tagging rare SNPs? So considering all questions of that type, you have to keep this fact about R in mind. Okay. Okay, this is one of my technical slides. I would just, perhaps if you're interested, you can think about later, but I'm just going to skip. Okay, so let me move on to my next topic, which is natural selection. So sometimes an allele frequency, let me talk about the forward time view and the backward time view. So the backward time view, which says we have a sample that we can go out and collect, and then it has some tree of ancestry that eventually reaches this one common ancestor. That, if you think about it, that's really in some sense the dual of the forward time view, right? The forward time view says that there's one allele that's present, or one allele copy that's present way back in the past, and it's possible through just random fluctuations at some point in the future for all of the genes to be descended from that one allele. That's really just the opposite ways of looking at the same thing, right? Okay. All right, so sometimes allele frequencies don't change just purely due to these random fluctuations. Sometimes they tend to head toward one boundary or the other. Okay. And such systematic change in allele frequency occurs in response to natural selection. Now, evolution by natural selection requires the following conditions. There are individual differences that affect fitness, fitness being the ability to survive and to reproduce. And some fraction of those genetically caused individual differences, in fact, some fraction of those individual differences are in fact genetic. So this basically means that the heritability of the trait is non-zero, right? Now, if these conditions are met, then those individuals who are better at surviving and reproducing pass on to their offspring the genes partly responsible for their success. And in this way, later generations become increasingly genetically and phenotypically more and more like the fittest members of earlier generations. Okay, so here's a picture I used to illustrate this. Suppose that initially the population is homogeneous for this green phenotype, but a mutation appears that confers this purple phenotype, and for example, for some reason this purple phenotype allows you to survive and reproduce better in the current environment. and assuming the environment doesn't change, in the next generation, we'll see slightly more of this allele conferring the purple phenotype. And then this process continues. The greens are not as good at surviving or reproducing. In the next generation, we see that the prevalence of the purple allele goes up even more. And if this continues, eventually, it could be that you have a fixation that's induced not by random fluctuations just happening to be absorbed at the opposite boundary, but just this kind of steady, systematic increase. All right. So in the previous slide, we saw how the frequency of the, let's call it the fitness plus allele, went from 1/7 to 7/7. This illustrates that natural selection is a systematic force that tends to send the allele frequency in one direction. However, it's often well, I guess always the case that genetic drift, which are these fluctuations that are unpredictable and can go in either direction, and natural selection, which ticks things upward in a consistent direction, these are always happening simultaneously. Okay, they're always happening simultaneously. So we have to keep that in mind. So I'm going to actually depict that in a more dynamic picture. So one thing to sort of understand what I'm about to show you is that these are pictures of the forward in time evolution of SNPs that are affecting, say, the same quantitative trait. Okay, and say we're in each case graphing the frequency of the plus allele, the trait enhancing allele, and suppose that selection is causing the mean of the trait to increase. Okay, so that means there should be a systematic tendency for the plus allele to head upward toward a frequency of 1. But we'll see that doesn't exactly always happen. So here we go, SNP1. We see all these random looking jitters. Okay, that time, oh, see, sometimes the fluctuations can take the allele the wrong way. But most of the time, it should go, oh, it almost went the right way, but then it reversed. Okay. So you're not safe, even if you're at 0.9.
All right, is this planned or you're randomly generating those?
I don't remember where I got this anymore. So, but yeah. Okay, so genetic drift, natural selection, they're always happening simultaneously. And in fact, for a quantitative trait, a quantitative trait to evolve in a predictable way actually relies on aggregation of the action of multiple SNPs, because at any given SNP, its evolution is quite erratic and can go even the opposite way of what selection would prefer. Okay, so genetic drift and selection, they always are acting simultaneously. Okay, so a question that arises is that, well, I can imagine that the undirected fluctuations will depend on the population size, whereas the directional tendency to do selection kind of is invariant. And so you might imagine, well, maybe there's some situations where selection overpowers drift and other situations vice versa. Okay. Now this question has a fairly precise theoretical answer, but instead of getting into that, I will try to get across some idea of the human case by giving you some quasi-realistic numbers. Okay, so earlier I alluded to the fact that the standard deviation of the allele frequency difference between Europeans and East Asians over SNPs that are common in Europeans, this is about 0.15. Okay, so you subtract, for example, East Asian from European, find the standard deviation of that across all SNPs, it's about 0.15. Okay, so that's the magnitude of what is mostly genetic drift. Now suppose that a quantitative trait is affected by, so that's basically what you see across the entire genome. There's a subset of the genome that affects some particular trait you're interested in. Suppose that there are 10,000 such SNPs, each with an allele frequency of 0.5. As we know, that's not realistic, but whatever, it makes the numbers easier. And the enhancing allele has an effect on the trait of 0.01. Okay, that is actually somewhat realistic. In fact, if you crunch the numbers, this produces a trait with a heritability of 0.5. Now, suppose that Europeans and East Asians differ respect to this trait for genetic evolutionary reasons by 2 standard deviations. Okay. Then we can ask the question, what will be the difference in the frequency of the enhancing allele at each of these causal SNPs? Okay. Now it turns out, perhaps surprisingly, that it is actually extremely small. It is 0.01. So in the population that has a higher mean, the frequency is going to be 0.51. And the population with a smaller mean is going to be 0.5. Okay. So this is an order of magnitude difference relative to the differences that we expect due to genetic drift. Now, I don't want this example to be too generalized. So this does not mean that drift just totally overwhelms natural selection, and that if we were trying to make cows yield more milk or whatever, this effort would be doomed to failure. The reason I in fact, bold all these words is that this particular conclusion about the relative strength of drift and selection depends a lot on the context. But anyway, the point I wanted to make now is that this does not mean that selection is wholly ineffective in a species with an effective population size of, say, 10,000 over the long term. There's an analogy here to statistical mechanics. So even if you were to follow the behavior of one gas molecule, it seems totally erratic and not obeying any lawful behavior whatsoever. But nevertheless, the aggregate behavior of all the gases in the box or whatever, if you're measuring certain aggregate quantities, such as how many of the particles are hitting the wall in a given interval of time and things like this, the aggregate can behave with undeviating regularity. Okay? So humans, when they apply artificial selection, for example, in domesticated species have even smaller effective sizes than humans, kind of paradoxically, kind of due to selection itself, because such a small fraction of the population is chosen to be the parents of the next generation. So there's just massive genetic drift and huge LD blocks in domesticated species. But nevertheless, if we graph how various trends with artificial selection go, what we see are these smooth curves with no stochastic fluctuations whatsoever. Okay, so it doesn't mean that selection doesn't work. However, what this fact about the relative balance of drift and selection at an individual common SNP means is that if polygenic adaptation has occurred, first of all, it requires great statistical power to detect it. For example, you would need like thousands of SNPs perhaps to beat down the standard error of the allele frequency difference to some manageable level. And number two, great care to distinguish any trend that you might observe from artifacts that actually, because of the smallness of the allele frequency difference, can be of comparable magnitude. And such artifacts can include things like ascertainment bias, where genetic drift in Europeans means it's easier to detect SNPs that have higher frequencies in Europeans versus other populations in a GWAS. Stratification in the original GWAS. All things I believe that you heard about here. Okay, moving on to my final topic, the impact of natural selection on patterns observed in GWAS. So earlier, we saw through coalescent theory, there are far more rare SNPs, the definition of rare being a bit depending on the context, but let's say less than 0.01, than common SNPs. And this is, follows from the 1 over p distribution. But does this mean that most of heritability is actually due to rare SNPs? Well, hard question to answer, but I would say not necessarily, and this is the reason why. So if the trait is standardized, then it turns out that the heritability of the trait is given by this particular equation up here, which I hope you've seen already. But even if you haven't, it's pretty intuitive, right? So it's saying heritability has contributions from all of the J SNPs, and each SNP's contribution has this factor p times 1 over p. That kind of makes sense, right? Because p times 1 minus p, that's the variance of a binomial random variable, and a SNP genotype is kind of like a binomial random variable. But you have two of them, so that's why the 2 is there. And then effect size squared. Okay, that's kind of intuitive why that would be there. Okay.
Is there an independence assumption?
Yes, that's why I have the approximation sign. This might not be exactly correct for reasons I think Loic will tell us about later today. Okay, so this equation derived by Fisher. So this factor p times 1 minus p, this already suggests that the fact that there are more rare SNPs is not going to be decisive, okay? Because that factor p times 1 minus p attains its maximal value for 0.5, and then it can become quite small once you're talking about, I mean, basically, it basically becomes 1 times a small number. So that can become very much smaller than, say, 0.5 times 0.5, which is 0.25. So basically, the contribution to heritability of rare SNPs can become dramatically suppressed by this p times 1 minus p factor. And so just the fact that there are more rare causal SNPs is not going to be decisive. Okay. Okay, so I'm going to talk about this a bit more. As I talk about this, it's going to be convenient to note that this factor p times 1 minus p, actually, it does not matter which allele you call ancestral or derived. So for example, suppose that derived is 0.9, ancestral is 0.1. But actually, we're mistaken. Derived and ancestral have been miscalled, which can actually happen for high DAF SNPs. But it doesn't matter because p times 1 minus p is the same, even if you switch the labels. So that means that actually, for temporarily, we can forget about derived and ancestral and just think about minor allele frequency. Minor allele frequency just being the smaller of the derived or the ancestral allele frequency. So if it's 0.9, 0.1, MAF is 0.1. Yep. Yep.
So I just want to understand a little bit about the rare SNPs because
Yep.
my understanding is that a lot of the GWAS essentially are powered only to get the common SNPs, right?
Yep.
And so in the case, you're defining heritability as related to the variance.
Yeah.
But if you imagine variance is somewhat insensitive sometimes, especially given the size, to outliers. And for a rare variant to, say, cause a single sample to be an outlier in the distribution, then it will have a big effect size, but it won't contribute much to the variance.
Yes, that's correct. Yep.
So in that sense, so it is possible for a rare variant to have, a spike in effect size, but still not contribute to the heritability so much.
I think I'm going to be talking about that shortly. So hold on to your question until a bit later. Yep. Okay. All right. Oh, whoops. That's supposed to pop up later, but okay. So it turns out if P follows the distribution 1 over P, what does the distribution of MAF follow? Well, MAF, if you have a SNP with this probability, you satisfy the same MAF as SNPs with this this frequency. So basically, you just add those probabilities, and you get 1 over P times 1 over P. Okay. So therefore, the contribution to heritability of all SNPs with a given minor allele frequency is given by this. The number of SNPs that have that minor allele frequency times the average contribution to heritability of those SNPs. Okay? So the number of SNPs at that MAF, that's proportional to the probability that a SNP has that MAF, and so that's 1 over this. And then we just saw that the contribution to heritability of a SNP with that MAF has to be this. Now notice something that's rather beautiful and unexpected. There's an exact cancellation here. This exactly cancels this. And so basically we're left with the contribution to heritability being proportional to just the average square of the effect size. Okay? Now further suppose that the trait is neutral with respect to fitness. So neutral means whether you're tall or short, if we're talking about height, that this has no impact on fitness. And furthermore, that mutations that have an effect on height have no side effects that have some kind of effect on fitness as well. Now if we assume that, this means that all mutations, regardless of their effects on height, are equally likely to drift upward to any given MAF. So if a thing has a 6-inch effect on height versus 1/6 of an effect on height, it doesn't matter as far as the likelihood of drifting to any given frequency. It's all the same. Okay? So that means that this factor of the effect size squared that's left after the cancellation, this is expected to be the same regardless of the MAF. Okay? And so that can be canceled as well. Okay? And so this leads to the prediction that MAF bends of equal length make equal contributions to heritability. Okay? So rather loosely, if we could put it this way, I add up all the heritability of contributed by SNPs that have MAFs within this interval. And as long as I keep the interval length the same, all of these heritability contributions are exactly equal under the neutral theory prediction. Okay? All right. So is this also magically obeyed by empirical genetic data? Actually, it turns out not. This is, in fact, not observed. For example, the same actually preprint I referred to earlier, Wingstein, this was actually not, the purpose of the study was not counting SNPs. This was to use GREML to partition heritability among different MAF intervals. So if the neutral theory prediction holds, all these data points should lie on this line, okay? So if I go to point 1, point 1 is 20% of the MAF interval, so that should explain 20% of the heritability. And then the next interval, this is like a cumulative heritability on the y-axis. And so the next interval should add exactly as much. So up to point 2, we should be at 40% of the heritability. So all of these points should lie exactly on this line. But when we actually empirically use GREML to partition the heritability for height and BMI, we see a deviation where immediately the first MAF bin, 0 to 0.1, explains 50% of the heritability, which is more than twice as much as what you would expect under the neutral theory prediction. Okay. So what does that mean? That means that mutations with a smaller effect are more likely to reach a higher MAF. Mutations with a smaller effect. Basically, mutations with a large effect are not actually likely to drift up here, and so they're sort of pinned down here. And that's why they explain more heritability than you would expect from the neutral theory prediction. Okay. Here's an example for a behavioral trait from Evans et al., where they did a similar thing, partitioning the heritability of IQ among different MAF intervals. They partitioned by LD as well. That can also be explained evolutionarily, but I'm not going to get into that. I mean, basically, I got this from the supplementary table and collapsed a bunch of categories, but 0.01 to 0.5, that's by far most of the NAF interval. That should account for most of the heritability. But instead of explaining most, you see the heritability actually looks more or less equally divided between basically this huge range and then this tiny range of MAF. Again, it appears that mutations with large effects on IQ are sort of pinned down to a low frequency by selection. For some reason, selection does not like these mutations with large effects on IQs. And therefore, the low MAF bin makes more of a contribution to heritability than the neutral theory predicts. Okay. So basically, can we empirically see this implication or this deduction that that mutations of large effect are pinned down to low frequency by selection? Well, it turns out that the answer is yes. So a number of years ago, Catherine Kemper and her colleagues came up with this very nice plot. Basically, on the x-axis, we have allele frequency. On the y-axis, we have the absolute effect of that mutation on height. So a lot of these things over here are mutations that are known to cause dwarfism or gigantism or Marfan syndrome or one of these things. Whereas over here, we have all the GWAS effects known at that time. Okay. So I slapped the citation here because there's actually nothing here. So there's nothing to cover up. Probably there is stuff here, but there's absolutely no statistical power to detect GWAS effect sizes that also have this massive p times 1 minus p suppression of the heritability. So there is stuff here, but it's impossible to detect, so why not just slap the citation? But the emptiness of this region is quite telling. These are large effects, or even intermediate effects in some sense, of common frequency. These would surely have been detected by now in GWAS, and the fact that we have not means that they definitely do not exist. Okay. Basically, So basically, drift becomes relatively less important as the effect of the allele increases, okay? In fact, a tenfold increase in the alleles per allele effect size can basically take it out of a regime where sort of drift dominates to one where selection dominates, okay? And so we can see that basically, why do these SNPs reach a high frequency? Well, their effects are so small that this 0.01 versus 0.15 comparison I was illustrating earlier applies to these SNPs, and therefore drift dominates, and despite whatever small deleterious effects they might have, they can drift up to a high frequency. However, once a mutation's effect on height gets up to like 0.10, 1, or even several factors of 1 for like something like Marfan syndrome, we can see that the balance reverses. Now selection totally predominates over drift, and these things are more or less deterministically pinned to a low frequency. I mean, there's no stochastic wiggles that can take these all the way up to here. Okay, so selection is the explanation of all these patterns, the heritability partition, and the relationship between allele frequency and effect size. But what explains this apparently ubiquitous signature? Yep.
When
Yep.
you get down to the very rare things,
Yep.
I wondered if most of the things that have huge effects that
Yep.
are quite rare, don't they have to mostly survive by being recessive? Because the total elimination would go much quicker if they were a dominant bad gene than if they're a recessive bad gene. So for those things on the left upper thing,
Yeah.
it's going to have to start mattering whether they're dominant or recessive.
Yeah, but my initial reaction is to say that, so if in one copy, like for example, in the first person who carries that mutation, there's essentially no effect. It's recessive. That must mean the selection against the phenotype, once it actually manifests, must be even stronger than you might otherwise think, because the recessivity, that means the lack of any phenotypic effect in one copy means that it's essentially neutral for a long time until homozygotes for the double mutants start to appear, and then selection kicks in. And so basically that seems like that would give a lot of scope for the mutation to just drift upward to a relatively high frequency. But that doesn't even happen, so there are several possible explanations for that, one being that, I mean, selection is really, really strong.
So you're kind of, one of the things you're saying is if it's recessive, then the beta that you would measure would be a lot lower.
Yeah.
So, I mean, it might be messed up in some way, but in terms of this kind of diagram, the beta being really big is partly a function of how horrible it is and partly a function of whether it's a recessive or dominant gene.
Yeah. I mean, the general point is true, yeah. Especially because non-additivity becomes more prominent as the effect size increases. For these rare mutations, a very powerful effect. You start after thinking about whether they're dominant or recessive and so on.
Although,
Yep.
I mean, for something this rare, like, what are the chances that you're going to find someone with 2 copies of the mutation, right?
Mhm.
Like, in a sample of 1 million people, you'd maybe have 1. It seems like it'd be really hard to test dominance or recessive in this case, right?
Well, they know it. I mean, I believe for some mutations that cause dwarfism, they're known to be dominant or to be recessive.
Sort of mating, and then you'll have.
Or inbreeding, actually.
Or inbreeding.
Yeah. Okay. So I was talking about where does all this ubiquitous selection come from, and also, this is, as I've suggested with switching from height and BMI to IQ. This is a very general phenomenon. This curve relating allele frequency to effect size seems to apply for just about any trait. So what explains this ubiquitous selection? Well, one clue comes from the fact that it appears to be insensitive to sign. Oh, actually, a point I forgot to make. So the heritability of IQ, according to G. Remmel in that study, was 0.39. Twin study suggests it's higher than that. But the mere fact that the heritability of a typical trait is far short of 1, that by itself also is evidence of natural selection. Because if mutational, they do these studies where they start with a pure inbred line of some organism, and then so pure inbred means everyone's genetically the same. So that means the heritability of the trait has to be 0. And then they basically allow mutations to freely accumulate, trying to keep the population at a small enough size that drift is much more powerful than selection. And they just see how much the heritability increases. And that's where that 0.001 number comes from. If there was no selection opposing these new mutations that are causing this heritability to click upward, it would not take a very long time in terms of evolutionary timescales for the heritability of the trait to be very close to 1. But the fact that hardly any traits have a heritability close to 1 suggests that most of this 0.001 that comes in is not destined to remain around for long. That's not just stochastic fluctuations that selection wants them to go away. And that by itself is evidence that the action of selection is ubiquitous. Okay, so the insensitivity to sign, back to now. This means that if we look at dwarfism, gigantism, and so on, there are basically no mutations of large effect on height at a high frequency, regardless of the sign. Okay, we don't see any height increasing or height decreasing alleles of large effect that have a frequency of, say, above 0.01. Okay. So this is actually the signature of stabilizing selection. So normally when we think about natural selection, we think about selection for giraffes' necks to become longer, human brains to become larger, gazelles to run more swiftly, and so on. And so this is called directional selection. But actually, a form of selection that's probably more common is stabilizing selection. This kind of selection is a selection for the current average, and basically results in a reduction of fitness for individuals who are far from the mean in either direction. Okay, so we have Yao Ming here suggesting that being extremely tall is not so great. Being short is, we can see from the data, is not so good for your fitness either. So basically, stabilizing selection, it turns out, penalizes mutations of either sign. Okay. Sometimes people have the intuition that that stabilizing selection should actually act to maintain the existing genetic variation, kind of like heterozygote advantage with a single SNP. But it turns out that that intuition is actually incorrect. That can happen with 1 SNP or 2 SNPs, but if a trait is affected by dozens, hundreds of thousands of SNPs, then what stabilizing selection does, it tries to drive out all existing genetic variation. Okay, the intuition is it wants everyone to be the same. It wants this distribution of height to go away and for everyone to be bunched up on one point. Okay. Now, since most traits probably do have some optimal intermediate value, in the fossil record we see few examples of phenotypic averages just kind of following a random walk over time. In fact, for some species, trilobites, coelacanths, they can look almost exactly the same over tens of millions of years. Stabilizing selection is almost certainly a ubiquitous force. Now, another source of ubiquitous selection is something called pleiotropy, which I believe is a term we've already introduced. Pleiotropy just means that a mutation with an effect on one trait also has effects on other traits that we might consider to be distinct from the first trait in some sense. Okay. Now, if we consider a mutation of large effect on some focal trait, even if there's no stabilizing selection, and even if maybe there's even directional selection such that in the absence of any other forces, that mutation of large effect would actually be favored, its side effects on other traits are probably disfavored. They're probably actually harmful. One way to think about that or rationalize that is that we think that most mutations are random. That is, there's no inherent tendency for mutations to go one way rather than the other with respect to phenotypes. So when we think about fitness itself, probably most random changes of large magnitude are deleterious. Okay? If I randomly change some component of this machine right here, most of those random changes are probably not going to improve its CPU speed or its RAM or its disk space or what have you. Okay. So most random changes are probably harmful, and so probably as the effect of a mutation on a trait that you're focusing on increases, its pleiotropic effects probably tend to become more harmful. Okay. All right, so I think this suffices to explain all these phenomena, the heritability partition, the relationship between allele frequency and effect size. Yep, there's a question. Yeah.
Can you say something about, it seems like the mechanism is inconsistent with the idea. How does the population keep track of the index of the variability in the population? When you say there's a driving force to keep the variance low in the population, but the mechanism does not respect anything in the population. So I guess, are you talking about something?
I mean, basically, think of it as if you're extremely tall and you do not survive and reproduce. The population loses a bunch of plus alleles. You're extremely short. You likewise fail to survive and reproduce. And basically, the way I think of it is, like, suppose you have some distribution of height. The tails have lower fitness, so they do not survive and reproduce as well. And so in the absence of any other forces, such as mutation, in the next generation, the distribution of height should be narrower, right? Because the tails did not reproduce as well as the people close to the optimum. And then you iterate that over generations, and eventually you're out of all genetic variation.
But that sounds like the second kind of selection. Maybe I don't understand the difference between the.
So stabilizing selection is something that's directly related to the trait itself, like I've just been talking about. That is, it's like, for example, there was a study that some psychologists did of speed dating in New York, where they looked at various characteristics and looked at how well they correlated how many speed dates someone would get. And they saw that males who are 5'3" need to earn $300,000 more per year to get as many speed dates as males who are of the average height. Okay, so that's like a direct, presumably causal effect of height on your fitness. Pleiotropy means that these same SNPs affecting height affect other things, and if most of those side effects are deleterious, that's another source of selection against them as well. Actually, empirically, these would be hard to distinguish, actually. If you looked at speed dating data of this kind, how would you know that the 5'3" guy didn't get a lot of speed dates? Because women just don't like men who are short, per se, or because being short, I don't know, is some kind of indicator of having lots of pleiotropic bad qualities? That would be an interesting, yeah. So empirically, actually, it would not be so easy to distinguish the two, but conceptually, I think they can be distinguished. Yeah.
From your example, I get a sense that the first one has a social mechanism associated with it.
Yeah.
We drove the small people to hurt their knees in the NBA league, or there's some publicly observable thing about being short, or something along this line. And this is consistent with this idea, but is that how you think about it? That there is a social component? Maybe sexual selection or something along those lines. It sounds more social. Is that what you mean when you talk about the stabilizing force as opposed to the like intrinsically?
I mean, by stabilizing selection, all I mean is that even if in the absence of pleiotropy, there is some optimal value of that particular trait, such that if you have too much of it, that's bad, and if you have too little of it, that's bad. Pleiotropy would mean that even if selection per se did not care about how tall the individuals in the population were, that these mutations would tend to have side effects, and those side effects, if they're deleterious, would explain why variants of large effect on height tend not to become common. Okay, let's go around the room here.
Also, in the case of Yao Ming, I mean, don't you think Yao Ming got so much injuries because he was so good at basketball? In a sense that, like Lionel Messi, he's not extremely tall or extremely short, or many other good players. Usually, excellent players get more injuries because they're excellent.
I sort of doubt that as the explanation, because we have a lot of players. I mean, Yao Ming didn't even play that many minutes, well, because of his injuries. But other players who played a lot more than he did that didn't have all these series of injuries to his ankles and to his knees and so on. And basically, I've heard from tall people that say that, yeah, don't be fooled by what you see on TV. We actually tend to be somewhat clumsy and have all these physical problems. So, yeah.
I was actually wondering,
Yep.
one of the things that, so stabilizing selection seems to be counter to the idea of evolution itself, right? Because evolution kind of gives this notion that you're actually deviating constantly, consistently away from these mean traits.
Yeah.
And that you're branching up. You have various branch points. And so, I mean, one possible explanation is that, I guess, stabilizing selection assumes the existence of a consistent environmental condition.
Yes, it assumes that the environment with respect to that trait is constant, for a long time. Yeah.
So because human beings, like the speed dating example, I know feels like we have complex societies with multiple kind of contexts in which those things work. But I mean, I'm just thinking, like, I guess your example is for stabilizing selection is like in say mouse lines or whatever.
Yeah.
So their assumption of environmental conditions being stable is that kind of like, is that correct or what is it?
There's actually kind of a debate about this. Some people think that that it would be typical in nature for the optimum to suddenly change, to become far away from where the population is presently located. And this would mean that directional selection would be happening all the time. And other people who think, well, no, the environment is probably constant. The constancy of the environment probably prevails over a pretty long time span. I would say that probably there's no general statement you can make about this that would be satisfying. So we seem to have examples from the fossil record where species like coelacanths, which are still alive, and you can find them in the waters of Africa, that as far as we know, look exactly the same as their fossil ancestors of 100 million years ago. There's some other species like this, horseshoe crabs. And then sometimes things happen, like asteroids or comets crash into the Earth, and then these small shrew-like animals that were around back then suddenly radiated and became like all these different kinds of mammals, including some that fly and some that swim. And so I don't know. I mean, sometimes people want a general, typically applicable answer to this question, so they could plug the answer into some other question, like about why is there sexual reproduction or something like that. But my first reaction is to say, it probably just depends. Some species, like humans, because we in fact change our environment so dramatically with agriculture and so on, probably evolve quite rapidly and are more often than normal subject to directional selection, whereas other species, the deep sea, for example, appear to inhabit a more constant environment, and there seems to be no point in making some kind of general statement.
So I think it's also likely they both happen at the same time.
Mm-hmm.
So if the optimal is just a little bit off the current mean, you're still going to get apparent stabilizing selection, but the mean can still change over time.
Mm-hmm. Yep. Yeah. Directional selection actually probably works that way. Instead of like this exponential increase where the taller you are, the better off, it's probably, there's still some kind of mound-shaped fitness function, and the optimum just shifts a little bit in each generation. Yeah.
I just wanted to add this example about the head circumference,
Yeah.
which has to match somehow the hip circumference of the mothers. If you think about being a too small baby,
Yeah.
probably not good for fitness, but
Yep.
being too large baby, have to adapt to how also the hip of the mothers will increase. So I guess that will
Yep.
play in both directions.
Mm-hmm. Okay, so the last slide. So basically, all this discussion leads to a question. For example, if there was no selection, the heritability of most traits would be pretty close to 1. However, we don't empirically observe that. We observe typically moderate heritabilities. But it turns out that even for under the strength of selection that we observe, and various other complicated things about, like, well, how strong must the selection be to keep all those height effects of large effect down to a low frequency and so on, it's actually possible to try to estimate how many sites are there in the human genome that can affect a given trait, such as, for example, height, or schizophrenia liability, or whatever. So there was a paper in Nature Genetics that used all these simulations to try to estimate this for type 2 diabetes, and they came up with a number of 1.2 million. So the geneticist Michael Goddard has kind of done more back-of-the-envelope calculations about this that I find a little more appealing. So the number he comes up with, basically he takes all this data about the heritability of the trait and what GWAS are showing about the trait and what these studies of mutations that are very rare are telling us about effect sizes and so on. And the mutational heritability from these other kinds of studies, and tries to put them all together and tries to come up with a back-of-the-envelope estimate of how many sites in the human genome, if they were to be polymorphic, would actually affect height, for example. And the number that he comes up with is about 10 million. Okay, 10 million. I find that to be actually staggering. That would have seemed like science fiction a decade ago, but now that's what we know now. Okay. Now, actually, for a long time in population genetics, people raised the question. This was back when people thought this number might be more like 5 or 20 or something like that. And you can even find video of Eric Lander in his OpenCourseWare site talking about, you know, now that we've sequenced the human genome, we are going to find the 5 SNPs that affect how good you are at playing chess and things like this. This has been all turned upside down with this. And so in one of his papers about this, Michael Goddard raises the question, so this is the answer to why heritability is not 0. It's just that there's so many sites that could contribute to it that inevitably you're going to have lots of mutations creating all these SNPs and so on. And the reason he says, I find this to be an interesting quote in the paper. Most authors have ignored this conclusion, perhaps because they regarded it to be too extreme to be realistic. The conclusion of this paper is that it is not extreme enough. Okay. All right, so for you to look at later, I have a checklist of questions where you can just ask yourself, do I know the answer to this? And if you do, that means you've pretty well absorbed the content of the lecture. And I also have some suggestions for further reading if you're interested in what I've been talking about. I actually have this book with me. I brought it here. This was the one book about population genetics that I packed, which I guess says something about that book. Maybe just that it's light. Some of these other books are not so light. This is actually physically 1,500 pages, the largest, heaviest book I own. And here's some of the classics by the founding fathers of population genetics and the first children, Gustave Mallico and Modu Kimura. I would say that out of all these books, this book is my favorite. This would be one to take on the desert island. Okay, I finished exactly on time, I believe. Okay, thank you. All right.
Any last questions?
Miles? Yeah.
I'm trying to think of the difference between the stabilizing selection and the pleiotropy, and it's related to another thing, possibly. So if it's really stabilizing selection and you have a hill, then it would be beta squared that would measure how deleterious it was.
Yes, you're right. Yes. Stabilizing selection, the selection coefficient goes with beta squared. Yep.
And possibly the pleiotropy could be more
pretty good, Miles.
proportional
Okay.
to the absolute value, because it's, with the
Well, it's not, we can't say a priori. We can't say. It depends on exactly how.
if it were
People have tried to derive that from Fisher's sphere model, and that will give you an answer, but we don't really know a priori. Yeah.
If it were like beta squared, then it's not really distinguishable from, it's just another form of stabilizing selection.
Yes, empirically, if the pleiotropy was related to the effect on the focal trait as such that the pleiotropic selection coefficient went as beta squared, and then you tried to infer all this from just looking at how selected against mutations affecting that trait are, what would happen is you would just think stabilizing selection, direct stabilizing selection, causally emanating from the trait itself was stronger than it actually is. And yeah.
So when you show that graph that show the population and then the division in
Yeah.
different groups.
Yeah.
And one question is about the non-randomness of this separation, right? So
Uh-huh.
the individual that separate from the main population, they will have share similar characteristics, and possibly these characteristics might be driven by natural selection before the actually separation.
Could be, but as far as I'm aware, models that try to say that, for example, that American Indians are just a random sample of some population, 60% that resembled East Asians, and then 40% this population called the ancient Northeast Asians. That these work perfectly fine. And also, the point I made about 0.01 versus 0.15 would apply, especially back then. So all these patterns, such as numbers of SNPs, allele frequencies, LD blocks, I'm pretty confident they are primarily due to drift rather than some kind of selection for a tendency to migrate or something like that. Yep.
Super basic question from way back at the start. I feel like
Yep.
but so when you're talking about the monomorphic site frequencies, you said it was 1/2n. Why isn't 1/2n minus 1? Assuming a degree of freedom in observing your first allele.
So all I'm saying there is that once a mutation first appears, its frequency just has to start at 1/2n. Because that mutation, suppose I'm carrying it, that's the only mutation that exists in the whole population. And then the denominator is 2n because if the population size is n, each of us carries 2 bits of DNA at any given site, one inherited from each parent. And so the number of sequences, I guess, is twice the population size.
Minus the one that you know that is already there. I appreciate that it doesn't make a difference, I guess, when the population size is larger.
Yeah, I'm not sure what you're getting at, but yeah, you're right. If it's up to 1,000, 10,000, yeah, minus 1, it's not going to.
Okay, so we'll continue at 11:20.